Welcome everyone, I am János Juhász and this is the data visualization course. In this section we will have a small tutorial about Jalview. Here you can see Jalview. If you opened it and closed all the pop-up windows, you see something like that. The first step is to get an alignment. You can do it with File, Input Alignment, you can insert it manually, you can get it from the internet, or you can get it from a file. Here I choose this Jalview sequences FASTA file. You will get this file. You can find this file at the course website. Jalview can read different kind of sequence files. So, for example, FASTA files, different extension can understand different extensions of FASTA file, Stockholm format alignment file, and so on. So the point of that, you need a sequence file to load. Okay, so let's open this file. Here, every results of every calculation will appear in different window in the Jalview. So if you work a lot with that, you will have a lot of windows inside that main window. Okay, so we see that we have 5 sequences. These are the names of the sequences, and these are the amino acid sequence. We see these are spike protein sequences from different coronavirus. So from coronavirus strains identified in different organisms. So, for example, this is from human. This is from also human, but a different coronavirus strain. This is from civet cat. This is from bovine, and this is from a bat. But they are all sequences of the spike protein of those coronaviruses. And we can see the sequences. Here in the conservation plot, we see that there are some similarities, but they are far from identical. But it's not surprising, because these are just sequences, so they are not aligned yet. These are just the bare sequences written on the top of each other. So let's do some alignment. You can do it with the web services. Then alignment. You can choose from different kind of aligners. Let's do the T-Coffee aligner with the default parameters. Then it will contact to the server that does the multiple alignments, and the server soon gives us back the result. The result, so technically the alignment itself, appears in a new window here. Appears in a new window here, and now we can see the alignment. We can see that there are gaps in each sequences. So, and with these gaps, the sequences match better with each other. From the consensus plot, we see that we have much more high conserved regions than we had at the case of the original sequences, when we just wrote the original sequences on the top of each other. See, there are some quite well matched regions. There are regions that appear in only one of the sequences and totally missing from the others, and so on. We see that, for example, this region is quite conserved, because there are a lot of high conservation point. But for example, this middle region is much less conserved. We can make it more clear by adding some color to the multiple alignment. So for example, if we have this Zappo color scheme, we see that the identical and similar amino acids are colored with the same color. So here, for these high conserved regions, we saw there are a lot of columns with the identical color. So they also have to visualize, these colors also helps to visualize the matching regions. If we would color just the sequences without an alignment, we would see much less conservation here. But after the alignment, the similar regions appear. Here we can use other color scheme with similar meaning, or we can color by, for example, the percentage of identity. Here, the more conserved regions will have darker blue color, and the less conserved regions will have lighter color. Here, the more identical and less identical regions will have more blue color, and the less identical regions will have lighter colors. So here, these dark blue regions are those where all the 5 sequences have the same amino acid at the same position. You can, for example, color the sequences based on their hydrophobicity value. For example, if you assign a hydrophobicity value for each amino acid, you can make a coloring based on that, or based on the buried index, so how likely that they are buried inside the protein with the 3D structure. And with this buried index is corresponding to the 3D structure of the protein. And here, based on these buried indexes, there are pretty nice consensus between the sequences. It could mean that their 3-dimensional structures could be related. Let's back to the Zappo color now. Okay, so with this format tab, you can decide what you want to see in the alignment. For example, you would like to see the text or the boxes or not. With this annotation field, you can show or hide the annotation visualizations, the conservations, the qualities, the consensus. Or you can add additional annotations. So for example, we can have this sequence logo annotation. And if we add this sequence logo annotation, then we will see this so-called sequence logo plot here at the consensus field. And it means that it's better in this way. It means that here only the most frequent amino acid appears in each position, and if there isn't the most frequent one, it is denoted by this plus. By this sequence, but the sequence logo plot also draws the most sequence amino acid or nucleic acid in case of DNA or RNA to the corresponding position, and it draws it with a big letter if it's very conserved. It draws it to many smaller letters if it's less conserved. So for example, here there is 100% M, so there is a big M in this sequence logo plot. Here at position 3, which is much less conserved, we have 40% I, 40% V and 20% L. So here it's a more variable position. Here the letters also appear, but with smaller sizes. And in the pure consensus sequence, there is nothing here. Here also there's 100% P, while here there is only 20% L. The other cases there is a gap here. You can make it also normalized, where the letters are written at bigger. So for example, here if you move your cursor here, you see all the letters, but they also have a bit bigger size, so maybe they are a bit readable, a bit better. So here in the sequence logo plot, all the possible amino acids appear. All the possible amino acids in a position appear. Bigger letters mean more likely amino acid at that position based on the multiple alignment, and smaller letters mean more rare amino acids at that position. So sometimes the sequence logo plot are also used to visualize multiple alignment. So a plot like this, you cannot do calculations with a plot like this. You can do calculation with the numbers, but for visualization, it could be quite useful to represent a multiple alignment. Again, these are also these are more detailed representations, but they are still aggregations. So because they say what amino acids could appear in these positions, but it doesn't say in which sequence. So the full data can be found in the multiple alignment itself and with the help of the colorings. But these are all aggregations. They are all concise descriptions. And with these descriptions, maybe they are a bit better visible, but you always lose details, lose information with these visualizations, because you don't know which are the sequences with the T, which are the sequences with the G, which are the sequences with the V, for example. Okay, but the sequence logo plot is also a quite useful tool, and the Jalview can do it. With the view, you can show sequence features, hide sequence features, sequence IDs, and so on. With the select, you can select a subset of sequences. So for example, I can select this sequence and this sequence and this sequence and this sequence, and then work only with that. So for example, if I have 10 sequences and after the alignment it turns out that only 5 is interesting, then I can select that 5 sequence and do the visualizations and the calculations with that 5 sequence later on. You can select all, deselect all, invert selection, create sequence groups, and so on. You can also edit the alignment. You can remove gaps you can remove positions from the left or the right, remove totally empty columns because if you import an alignment, it would contain totally empty columns. You can remove them. You can remove gaps, remove redundancies. So you can also edit the alignment itself. You can export the images as well. You can export it in different file formats. Some of them are vector-based formats. Vector-based formats are invariant for increasing or decreasing the image, so you won't lose image quality. You can export it HTML through the web, or PNG format, which is a pixel-based format. If you you can open it with more programs, but if you enlarge it, you can lose quality of the image. You can lose quality of the image if it's not high resolution enough. So it based. So if you, for example, if you can open vector-based images, so if you have some software tool opening vector-based images, you can download it in that format, and then you don't have to care about image quality. If you download it in a pixel-based format, then you will have to care about which size you would like to show, in which size you would like to show the image. If I download it like that, it will create an image from the alignment, and I can download it later. And if it's ready, I can download it. Okay, so for it works. Okay, I save that image. And let's see what kind of calculations I can do with my multiple alignment. I can sort it based on, for example, sequence ID length, pairwise similarity. If I sort them with pairwise similarity, hopefully the more similar sequences will be close to each other, so this coloring will be even more informative. And it will help me to identify which sequences are closer to each other. From here, it sees that this last sequence, which was the bovine sequence, maybe this is the most distant from the others. It has some sequence regions totally missing from the others in this middle part, if you see. You can also do tree or principal component analysis with your sequences. You can make the tree based on different sequence matrices, and you can use different tree making algorithm. Now just use the most simple one. Here, this is the sequence tree. If you can select some sequences in the tree, and you see that it gets selected here as well at the sequence list. And you can group them together just clicking at the plot, and it will draw this red vertical line, and sequences after that red from right to right from that vertical line will have the same color, and from the left they will have a different color. So with this, you can see that these are the two most similar sequences. These are these bluish ones, the civet cat and one of the human samples. Then with this green one, this bat is also quite similar. The most distant, yes, the most distant is this one, the bovine sequence. And here is the other human sequence with this pinkish color. So for that, you can identify which sequences are closer to each other. If you draw the principal component analysis, it will give a plot like this. The colors are the same. If you see, you can zoom to this picture and you can rotate it. And technically, sequences that are closest to each other should be the closest in this principal component analysis plot. Here, each dot corresponds to one sequence. Here, you can choose different dimensions. I will now choose the first dimension only. If I choose the first dimension only, it turns out that this white sequence, the bovine, is the most distant from the others. If I choose the second one, so if I choose the second one also, then it turns out that so yeah, based on the first dimension, these one are the closest to each other. Based on the second dimensions, these ones, the bat and the other human, are also close to each other. Yeah, so from these, for more sequences, this principal component analysis plot would be more informative because you would see groups of dots and sequences corresponding to that group of dots would be those who are more similar to each other. Okay. You can, as I mentioned, you can also assign three-dimensional structures for the sequences. For that, you can select a sequence, just clicking on them with right-click, and then click on this three-dimensional structure. There are no three-dimensional structure for this sequence. Then let's choose another. It can happen that for a sequence you don't have three-dimensional structure. In this case, you won't find any. For this, there are many possible three-dimensional structures. Let's choose one of them and click New View. In this case, a new window will appear with the three-dimensional structure of corresponding to that sequence, this Q3. This was the civet cat sequence. Here you can also zoom in and out to the sequence, and you can rotate it. And the coloring is the same as the coloring in the three-dimensional case is the same as the coloring at the plane sequence. But you see that there are other parts as well, these gray regions. It is because this is the structure of a whole protein, a protein built up from multiple subparts. So you can, for example, you can also save these images, but you can also select which side chain you want to see. Okay, so it was the first side chain. So let's only visualize the first side chain and delete all the rest. In this case, we see only the first side chain with the same coloring. You can see that there are some helical regions. There are some helical regions in the protein, and there are some beta sheet regions, and so on. You can change the coloring by hydrophobicity, for example, or to buried index, for example. So you can view sequences, the three-dimensional properties of sequences as well. Through the Jalview. So you can also see the charges and the cysteines inside the sequences and so on. you can also use other web services through the Jalview. For example, you can okay, we don't have any sequence selected. So you can, for example, try to predict protein disorder from the sequence. There are many tools for that. Let's use this IUPRED. Let's select all the sequences. So I selected all the sequences, and now from here I get, so I selected all the sequences, and now I can see the predicted protein disorder pattern of these 5 sequences based on the IUPRED protein disorder prediction. Here the high values are corresponding to the potentially disordered regions of this protein, while the low values in the bar graphs are corresponding to the regions that are likely to have some order. So if I color it based on the helix propensity, with these regions with the high and low disorderedness. So for example, these regions has very low disorderedness, these regions have higher disorderedness. So the helix property colors are also, if you see, are a bit different. You can also predict secondary structure elements. Let's use this tool for that. Here it opens on a new window. And here you can also set the color. You can see different, you can see the regions where some secondary structure elements, alpha helices or beta sheets are predicted in these sequences based on different parameters, different methods. So if we see that, for example, here, around the position 1000, and the position 1000, this huge beta sheets are predicted. And in this area, the disorderedness, it's also predicted to be quite low. So this region is probably a quite ordered region. The disorder prediction is low, and we can predict some strong secondary structure elements there. So these are all the calculations, so they are beyond of visualization, but these visualization tools can help us to understand not the sequences not only the sequences, but the group of sequences. You can do this kind of visualizations for independent sequences as well, but here with this, if you do it for a multiple alignment, you can inspect and you can catch more general patterns than viewing individual sequences. You can also save these files and later reopen them in Jalview or you can export them as images as we did before. Okay, so that was a brief introduction of Jalview, and thank you for your attention.